Usually when we talk about biological teamwork, we picture it like this perfectly choreographed dance. Two proteins find each other in the chaotic soup of a cell, they bind together, they do their job, and everything just runs smoothly.
Right, yeah. And the standard assumption for a long time has just been that if one of those proteins is mutated, the job just doesn't get done. The machinery stops and the cell suffers, a pretty predictable consequence.
And that makes intuitive sense. But to kick off this deep dive, we have a biological mystery that completely shatters that assumption. We are looking at a specific pair of proteins that normally work as a team. But when one is mutated, the other one, the leftover partner, doesn't just sit around helplessly.
No, it really doesn't. It actually goes completely rogue. It takes on a brand new, totally unauthorized job and aggressively starts building liver tumors.
Which is just terrifying. It's the ultimate story of a microscopic partner going off the rails.
Exactly. And what's so compelling about this is that it completely rewrites our understanding of certain rare diseases and, crucially, how we should be treating them.
Welcome to the Deep Dive. Today, we are unpacking a really dense, just fascinating 2025 paper from the journal Cell, Death, and Disease. We're exploring a totally novel pathway in a genetic disorder called Tuberous Sclerosis Complex, or TSC.
Yeah, and our mission here is to figure out how a lonely, leftover protein can physically rewire the metabolism of an entire organ.
So to really appreciate the weight of what these researchers found, we have to look at the baseline of TSC itself, right?
Yeah. Let's start there. So TSC is an autosomal dominant disorder. In practical terms, for you listening, that means you only need to inherit one mutated copy of a gene from either parent to actually develop the disease.
Right.
And this disease is caused by inactivating mutations in either the TSC1 gene or the TSC2 gene. For decades, the scientific consensus was fairly straightforward.
Which was that a mutation in either of those genes did basically the exact same thing, right?
Precisely. We thought it disrupted the TSC protein complex and just removed the brakes from a crucial protein called mTORC1.
Let's hover on mTORC1 for a second, because that is a heavyweight player in cellular biology. That is essentially the cell's master growth regulator. It's the engine that senses nutrients and tells the cell, okay, it's time to grow, divide, consume resources.
And that engine requires incredibly strict supervision. So under normal, healthy conditions, the proteins TSC1 and TSC2 bind tightly together. They also bring along a third helper protein called TBC1D7, and together they form the TSC complex.
Okay, so it's a three-part team.
Right. And the primary critical job of this assembled complex is to suppress mTORC1. They're basically the regulatory brake pedal.
I've always thought of it like a buddy cop system. TSC1 and TSC2 are these two strict, by-the-book cops assigned to guard the mTORC1 engine.
That's a great analogy, actually.
Yeah, and as long as they're partnered up and patrolling together, the engine just purrs along at a safe, normal speed. But if either cop gets mutated and disappears from the force, the brake is gone. The mTORC1 engine just revs completely out of control.
Which triggers a massive cascade of downstream effects. When mTORC1 is hyperactive, it increases the signaling of an enzyme called GSK3 beta.
Okay, alphabet soup warning.
I know. But without getting bogged down in the acronyms just yet, what you really need to know is that this specific enzyme cascade triggers an increase in glycogen synthesis.
And glycogen is just how our bodies store sugar.
Exactly. So with either mutation, the cell's engine runs hot, and it starts hoarding sugar.
Wait, I have to stop you there, because this is where the source data it wasn't adding up for me.
How so?
Well, if TSC1 and TSC2 do the exact same job, if they are just two halves of a brake pedal holding back mTORC1, then losing either one should just release the brake, right? A broken brake is a broken brake.
You would think so, yeah.
But the clinical data in this paper shows a massive discrepancy. Mice with TSC2 mutations develop severe kidney lesions 60% of the time. But in TSC1 mutant mice, it was only 30.8%.
Right, almost double.
Yeah. And for liver lesions, it was 20% versus just 7.7%. Why on earth is the liver hit so much harder when TSC2 is the broken gene? What are we missing here?
Well, that logical gap you're pointing out is the exact thread these researchers pulled on, and it led to the major breakthrough of this entire paper.
Oh, wow.
The severity of the disease isn't just about the missing cop. It is entirely about the leftover cop. If a patient has a TSC2 defect, there is no TSC2 protein. The complex can't form, so the brake on mTORC1 is gone.
Right.
But you still have a perfectly healthy, highly abundant amount of TSC1 protein just floating around the cell with absolutely no partner.
So we have this massive pool of what they call uncomplexed TSC1. If I'm tracking with you, the original problem, the one we've known about for years, is that we lose the brake on cellular growth.
Correct.
But the hidden, much worse problem is that this leftover TSC1 protein is now a free agent, just wandering around the cell.
Yes, exactly.
Let's do a quick recap for you listening at home to make sure we've got this foundation locked in. Point 1, TSC1 and TSC2 normally work together as a buddy cop team to inhibit the growth regulator, mTORC1.
Point 2. Mutations in either gene hyperactivate that mTORC1 engine, causing some baseline glycogen or sugar buildup.
And point 3, TSC2 mutations are clinically far worse because they leave behind this rogue agent, uncomplexed TSC1. So here's a question for you to think about as we move forward. Imagine you are that leftover cop, TSC1. You're suddenly without a partner. You don't just disappear. Where do you go, and what kind of unsupervised job do you find?
To find out, the researchers essentially tagged and tracked this uncomplexed TSC1, and they found it actually leaving the cytoplasm, the main fluid-filled body of the cell, and crossing the threshold straight into the nucleus.
Wow, into the vault. The nucleus is where all our DNA is highly protected and stored.
Right. And once TSC1 enters the nucleus, we cross over from basic cellular metabolism into the realm of epigenetics.
Epigenetics, the software that runs the hardware of our DNA. Because, every cell in your body has the exact same DNA hardware.
Yeah, but epigenetics are the chemical tags that tell a liver cell to act like a liver cell and a brain cell to act like a brain cell.
Right, to do the vital distinction.
So inside the nucleus, the cell uses proteins to manage those chemical tags. And one of these management proteins is called KDM5A. KDM5A is a histone demethylase.
Okay, let's visualize that for a second. Histones are essentially the physical spools that our DNA wraps around to stay organized. If the DNA is spooled up tight, it can't be read. If it's unspooled, the genes are exposed and active.
And the way the cell controls that spooling is through chemical tags. One of the most important tags is called H3K4ME3.
H3K4ME3.
Yeah, it's a mouthful. But you can think of H3K4ME3 as a bright green light for transcription. When those tags are present, it tells the cellular machinery, unspool this section and read this gene.
Okay, green means go.
Exactly. Now, KDM5A's normal day-to-day job is to act as an eraser. It patrols the DNA and actively removes those H3K4ME3 green light tags, which ensures that certain genes stay quiet and turned down.
And this is where the leftover cop goes totally rogue. Uncomplexed TSC1 wanders into the nucleus and physically interferes with KDM5A, right?
Yes. It doesn't just passively float by, it actively downregulates the eraser, stopping it from doing its job entirely.
How is it actually pulling that off? Is it destroying it?
It's essentially a physical blockade. The uncomplexed TSC1 binds in a way that suppresses the expression and the function of KDM5A. And if we look at the physical consequence of that blockade, it creates a super dangerous chain reaction.
Because the eraser is sidelined.
Exactly. Because the eraser is gone, those H3K4ME3 green light tags start to accumulate, they pile up relentlessly, and the researchers found they pile up at one very specific, highly dangerous location, the promoter region of a gene called MTTL3.
Okay, the promoter region is like the starting line of a gene. It's where the copying machinery attaches. So we have this massive pileup of green lights at the starting line of the METTL3 gene.
Right, and because the signal is so blindingly bright, transcription factors, which are the proteins that actually kick off the copying process, rush in. The paper specifically noted that transcription factors called TBP and ETS1 bind incredibly tightly to this promoter now.
It basically creates a perfect runaway storm. The eraser is gone, the green lights are permanently stuck on, and the transcription factors are locked in place. The volume dial on the METTL3 gene has been cranked to absolute maximum.
And the cell just starts churning out staggering amounts of the METTL3 protein.
What really blows my mind is how the researchers proved this was a unique behavior. Because they looked at the opposite scenario too, right? The TSC1 mutant mice.
Yeah, where TSC1 is missing and TSC2 is the leftover partner.
Right. And if you look at their nucleus, this cascade doesn't happen at all. In fact, MTKL3 expression in those mice actually drops. That completely validates your point. This volume-blasting behavior is a toxic gain of function, entirely unique to the uncomplexed TSC1 protein.
It fundamentally shifts how we view the pathology here. The broken brake pedal is bad, obviously, but the leftover piece of the brake pedal transforming into a switch that supercharges a completely different gene, that is what drives the severe disease.
Wow. Okay, time for another quick recap to the listener. Point 1. Uncomplexed TSC1 enters the nucleus and suppresses the eraser protein, KDM5A.
Point 2. Without the eraser, H3K4ME3 green light tags pile up on the METTL3 gene promoter.
And point 3. The MET-TL3 gene is kicked into overdrive. So the question you should be asking yourself now is, we've just turned up the volume on MET-TL3 to maximum, but what exactly is a MET-TL3 printing out that makes the liver so incredibly sick?
To answer that, we have to look at what MET-TL3 actually does. It operates on a totally different level of cellular management. It is what we call an M6A writer. This gets into the realm of RNA modification.
Which is such a fascinating area of biology. Let's break that down. Our DNA is transcribed into messenger RNA, or mRNA. That mRNA acts as the temporary blueprint, carrying the instructions from the nucleus out to the cell's factories to build proteins.
But those blueprints aren't just sent out as is. The cell can heavily modify that mRNA before it gets read. An M6A writer, like MET-TL3, adds a specific chemical tag, a methyl group, directly onto the messenger RNA.
So if the mRNA is a blueprint, MET-TL3 is basically the boss slapping a giant fluorescent urgent VIP sticky note right in the middle of it.
That visual captures it perfectly. And in this specific pathway, MET-TL3 isn't just tagging random blueprints, it highly targets the mRNA for a gene called GYS2, which stands for glycogen synthase 2.
Okay, but a sticky note doesn't accomplish anything unless someone on the factory floor actually reads it.
And that brings us to the reader protein, IGF2BP2.
Another acronym for the listener to juggle.
I know, but think of it this way. If APTL3 is the boss writing the urgent memo, IGF2BP2 is the aggressive factory manager who looks for those tags. When it sees the M6A tag on the GYS2 mRNA, it swoops in, grabs the blueprint, and aggressively stabilizes it.
So it prevents the cellular recycling machinery from shredding that mRNA.
Exactly.
So you have a blueprint that tells the cell to build GYS2. It's been marked as highly urgent, and the manager is physically shielding it from being thrown away, forcing the factory to read it over and over and over again. What is the physical result of that happening inside a liver cell?
The liver cells become completely flooded with the GYS2 protein. And since GYS2 is the primary enzyme responsible for synthesizing glycogen in the liver cells are forced into overdrive. They start hoarding glycogen to dangerously toxic levels.
It's an assembly line of excess that simply cannot be turned off.
Right. And the researchers provided incredible visual proof of this. They took mice that were 12 to 13 months old and used a technique called PAS staining.
PAS stands for periodic acid shift, right?
Yeah. It's a specific chemical dye that turns glycogen bright magenta. So imagine looking through a microscope at healthy liver tissue, which should be relatively clear, with some structure.
Okay.
When they looked at the livers of the TSC2 mutant mice, the tissue was just choked, almost entirely saturated with this vivid magenta dye.
It really highlights the sheer physical burden on the organ. But it goes beyond just harmless sugar storage, doesn't it?
It does. These heavily congested livers were actually developing hemangiomas. Hemangiomas are tumors made up of abnormal, wildly tangled blood vessels and smooth muscle cells.
And the researchers confirmed the makeup of these tumors by testing them for specific biological markers, right? CD31, which highlights the endothelial cells of blood vessels, and SMA, which highlights smooth muscle.
Yes, and here's the absolute smoking gun of the entire paper. When they overlaid the PAS staining with the tumor markers, the areas of the liver that had the absolute highest concentrations of magenta glycogen were the exact same areas where these tangled, severe tumors were erupting.
Wow. It forces us to completely rethink the relationship between metabolism and cancer. I mean, we often view tumors as these isolated runaway growths that just steal resources.
But in this case, the early-stage liver tumors actually rely on this pre-existing massive glycogen storage to initiate their growth. The glycogen isn't just the sad byproduct of a sick liver, it is the raw fuel for the tumor.
The rogue uncomplex TSC1 essentially built a massive fuel depot, and the tumor just uses it to ignite.
It is brilliantly devious.
Let's quickly summarize this crazy assembly line for you listening. Point 1. MET-TL3 tags GYS2 mRNA, and the manager, IGF2BP2, stabilizes it.
Point 2. That stabilized GYS2 creates massive, uncontrolled glycogen storage in the liver.
And point 3. This excess glycogen directly fuels the formation of severe liver tumors or hemangiomas. So think about this. If you are a doctor treating a TSC patient right now, and you know the standard treatment only targets the original mTORC1 engine, how do you address this newly discovered, entirely separate MTL3 glycogen pipeline?
That transition from mapping a pathology to actually altering clinical treatment is where this research becomes incredibly hopeful. It's a massive leap toward true precision medicine.
Let's examine the current standard of care. Right now, patients with TSC are typically given mTORC1 inhibitors, drugs like rapamycin or everolimus.
Right.
So if a patient with a TSC2 mutation is taking rapamycin, believing it's managing their disease, what is actually happening inside their liver?
They are essentially only fighting half the battle. Now, rapamycin is incredibly effective at fixing the original buddy cop problem. It acts as a chemical brake, jamming itself onto the hyperactive mTORC1 engine and slowing it back down to a normal baseline.
Okay, so the engine is fixed.
But remember that uncomplexed TSC1 going rogue in the nucleus, tackling the eraser, turning up the MET-TL3 dial. That is an entirely mTORC1 independent pathway.
So the drug is completely blind to it. Rapamycin fixes the engine, but it does absolutely nothing to stop the leftover cop from invading the nucleus.
And the data totally backs that up. When the researchers treated the TSC2 knockout mice with only rapamycin, the mTORC1 engine slowed down, which provided some systemic relief, sure, but their liver glycogen levels remained stubbornly high. The rogue pipeline was still wide open, dumping fuel into the liver.
This is where the researchers designed a really elegant intervention. They hypothesized a 1-2 punch. They decided to pair the standard rapamycin with a completely different experimental drug called STM-2457.
Right. STM-2457 is a highly specific small molecule designed to target and inhibit the MTL3 protein itself.
And the experimental design to prove this was super rigorous. They started with xenograft tumor models. To give you listening a mental picture of how they do this, they don't just rely on cells in a petri dish. They actually take these specific mutant human tumor cells and implant them under the skin of immunocompromised mice. This allows them to watch the actual physical tumor grow and react to the drugs in real time in a living circulatory system.
And in these living models, the results were striking. Giving the mice just rapamycin slowed the tumor growth slightly. Giving them just the MET-TL3 inhibitor, STM-2457, also helped slightly. But when they administered the two drugs together...
That's where the magic happened.
Exactly. The tumor shrank dramatically, and the toxic glycogen levels in the surrounding tissue completely normalized. By hitting both pathways simultaneously, they shut down both the hyperactive engine and the rogue fuel pipeline.
And they didn't even stop there. They wanted absolute genetic proof that this specific pipeline was the culprit. So they utilized an RAAV8 viral vector.
Which is such a cool technique.
It really is. They took a harmless virus, which naturally gravitates toward liver cells, and engineered it into a microscopic delivery vehicle. They load this virus with custom RNA instructions designed to silence either the METL3 gene or the G2 gene.
Right. And when they injected this into the tails of living mice, the virus traveled straight to the liver, delivered the silencing instructions, and successfully reversed the severe liver lesions.
It's basically like sending in a highly specialized microscopic SWAT team to quietly dismantle the rogue operation without harming the rest of the cell.
It represents the absolute gold standard of modern biological research. They identified a clinical anomaly, tracked it down to a completely novel genetic mechanism, proved every single step of the physical pathway, and then immediately demonstrated how to successfully drug it in living animals. For patients suffering from severe TSC2-related liver and kidney lesions, for whom rapamycin alone has just been a frustratingly incomplete treatment, this combination therapy represents a very real, tangible horizon of hope.
Let's do one final recap of this treatment paradigm. Point 1. Standard mTORC1 inhibitors like rapamycin don't fix the uncomplex TSC1 pathway.
Point 2. Combining rapamycin with a MET-TL3 inhibitor like STM-2457 fixes both pathways simultaneously.
And point 3. This combination drastically shrinks liver tumors and normalizes the toxic glycogen. It's a profound shift in perspective. We usually think of a gain-of-function mutation as a gene that's hyperactive from the start. But here, a broken system, a simple loss of a partner, created a leftover piece of cellular machinery that actively took on a toxic, completely unexpected new role.
It makes you wonder, how many other intractable, rare diseases are hiding a secret rogue agent behind a seemingly straightforward, broken protein complex.
Exactly.
And that's really the perfect question to leave our listeners with today, because if a protein like TSC1, which evolved over millions of years to do one highly specific, highly regulated job while bound to a partner, if it possesses this incredibly complex, pre-programmed ability to manipulate RNA methylation the moment it's left unsupervised,
Right.
What does that tell us about the hidden secondary lives of the millions of other proteins floating around in our bodies? Are they all just waiting for their partner to step away so they can start moonlighting?
A slightly terrifying, but ultimately beautiful thought about the sheer untamed complexity of human biology. Understanding that complexity is exactly why we do this. Thanks for joining us on this deep dive into the hidden lives of our cells. Keep questioning the consensus, and we'll see you next time.